A warm welcome to one and all, dear friends. Myself, Dr. Rashmi Joshi Sawalkar. Today, I am here to deliver my lecture on subject Zoology, and the title of the topic is Ribosome, Structure and Functions of Ribosomes. It is from the second unit of our syllabus, and let's see what are the functions and structure of ribosomes. So, what are ribosomes? Cell have tiny granular structures known as ribosomes. See friends, previously I explained you all the cell organelles which are present sorry, which are seen in the cell cytoplasm in prokaryotic and eukaryotic cells. The structures till now we have studied are Golgi complex, endoplasmic reticulum, then mitochondria, then nucleus, and now I am going to explain you the structure of ribosomes which are present on the endoplasmic reticulum, and hence the reticulum is called as rough endoplasmic reticulum due to the presence of these granules of ribosomes. So, cell have tiny granular structures known as ribosomes. Ribosomes are ribonucleoprotein particles. Ribosomes serves as workbenches with mRNA acting as the blueprint in the process of protein synthesis. A very important function is performed by ribosomes. That means protein synthesis is done by these minute structures which are very much important, and that is ribosome. So, ribosome structure is given here. See, two subunits are there. The large sized unit is called as large subunit and the small size subunit is small subunit, and this is the central protuberance. So, these two structures, large unit and small subunit, togetherly forms the entire structure of ribosome. So, this is all about the structure of ribosome. Now, let us see Palade. Who discovered ribosomes? Palade was the first person to study them in 1955. See here the scientist Palade, who invented or who first observed the structure in 1995. Now, how many number of ribosomes are present in the living cells? The number of ribosomes differs greatly. A rapid growing E. coli cell may have as many as 15,000 to 20,000 ribosomes, about 15% of the cell mass. It is here the E. coli structure, the bacterial Escherichia coli structure was given here. So, in E. coli bacteria, about 15,000 to 20,000 ribosomes are seen, that is the total mass of cell, 15% of the total mass of the E. coli cell. So, it can differ from cell to cell, it can vary from cell to cell. Now, types of ribosomes. So, matrix ribosomes, plasma membrane ribosomes. So, what are these matrix ribosomes? These synthesize proteins destined to remain within the cell. These synthesize proteins destined to remain within the cell. These ribosomes are present inside the cell and which synthesizes protein, which are only for the synthesis of protein. Now, plasma membrane ribosomes, these make protein for transport to the outside. Now, these ribosomes are for transportation. These transports the protein synthesized from the cell itself to the outside of the cell. So, these two types of ribosomes, matrix ribosomes and plasma membrane ribosomes. Now, domains of ribosomes. So, what are the domains of ribosomes? There are two domains of ribosomes, translation domain and exit domain. Let us see one by one. What is translation domain? The region responsible for translation is called the translational domain. Both subunits contribute to this domain, located in the upper half of the small subunit and in the associated areas of the large subunit. Now, in the ribosome structure, as I have explained you in the previous slide, that the larger subunit and the smaller subunit is seen. So, in the larger, both the units, that is the larger and smaller subunit togetherly combines and shows the presence of translational domain, associate areas in the larger subunit. It is present in the larger subunit of ribosome. Then exit domain, the growing peptide chain. It is, these are the domains where the protein synthesis takes place. So, translational domain, again it is a place where protein synthesis starts, and one by one amino acids binds here at the translational domain, and they are exited out and the protein chain is formed. So, the exit entry or exit place is called as the exit domain. The growing peptide chain emerges from the large subunit at the exit domain. This is located on the side of the subunit. So, these two are the domains which are present in the larger side or large subunit of the ribosomes. Now, dimensions of ribosomes. See, prokaryotic ribosomes are commonly called as 70S ribosomes. These are all the points which are asked in the objective exams generally. So, see carefully or listen carefully. Prokaryotic ribosomes are commonly called as 70S ribosomes. These have dimensions of about 14 to 15 nanometer by 20 nanometer in size. So, the dimensions you have to understand that in 70S ribosomes the dimensions are about 14 to 15 nanometer by 20 nanometer. Okay. The molecular weight of is approximately 2.7 million Daltons, that is 2.7 into 10 raised to power 6 Daltons. These are constructed of a 50S and 30S subunits. The 70S ribosomes is equals to 50S plus 30S subunit. This is the bifurcation, that how the 70S ribosome is made or formed. So, it is made up of 50S and 30S subunits, and we get the 70S ribosomes, which is the prokaryotic ribosomes. Now, ribosomes are not bounded by membrane. Prokaryotic ribosomes are smaller and less dense than eukaryotic ribosomes. Ribosomes are composed of two subunits, each of which consists of protein and a type of RNA called ribosomal RNA, that is rRNA. Ribosomes are composed of two subunits, as I have told before, that larger subunit and smaller subunit, each of which consists of protein. Each subunit consists of protein and a type of RNA. The RNA which is present in the subunits of ribosomes are called as ribosomal RNA or rRNA. So, these are present in the two subunits of the ribosome. See here in the diagram, prokaryotic 70S ribosome. This is the structure of 70S ribosome. This is the upper larger subunit, this is the lower subunit, and this outgrowth here is called as the protuberance, as I have explained you previously. So, here we will see the larger subunit. See, the larger subunit is 50S and the smaller subunit is 70S. These two subunits unite together to form the structure of prokaryotic 70S ribosomes. Now, in 50S subunits, here S stands for Swedberg unit. So, the invention of these units are done by the scientist Swedberg. So, in the honor of that scientist, the Swedberg S symbol is given to these units. So, 50S, that is Swedberg units, 30S, that is 30 Swedberg units. So, 50S consists of 23 S and 5 sRNAs, that is 34 proteins. So, total this amount of rRNA and proteins are present in these units. Here in this point I have explained that in the subunits of ribosomes, rRNA are present and proteins are present. So, how much amount of rRNA and protein are present in each subunit? So, see here in 50S subunit, that is in larger subunit, about 23 S and 5 sRNAs are present along with 34 proteins. Okay. Now, please learn these parameters, that in 50S ribosomes how much RNAs and how much proteins are present, and in 30S RNAs 60 sRNA and 21 proteins are present. Okay, so understand this bifurcation and keep in mind for further examination. Now, ribosomal subunits. Each subunit is constructed from 1 to 2 rRNA molecules and many polypeptides. 30S smaller subunit and 50S larger subunit. This is all the bifurcation about the larger subunit. Now, the S in 70S and similar values stands for Swedberg units. As I have explained you before, the faster a particle travels when centrifuged, the greater its Swedberg value or sedimentation coefficient. This is the sedimentation coefficient, for which S stands for sedimentation coefficient. When the ribosomes are centrifuged, fastly centrifuged, the greater its Swedberg value or sedimentation coefficient. The sedimentation coefficient is a function of a particle molecular weight, volume and shape. Heavier and more compact particles normally have larger Swedberg units. As the mass or the mass and the weight of the particle or ribosome is larger, the Swedberg number will also be larger and it will sediment faster. So, this is all about the S unit or Swedberg unit. Now, 30S subunit is smaller and has a molecular weight of 0.9 into 10 raised to power 6 Daltons. It is made up of 16 sRNA, rRNA and 21 polypeptide chains. So, these are the two units, small subunit, and this is the platform, this is the head region, this is the 30S structure of ribosome. Now, in 50S subunit, the 50S subunit is larger one and has a molecular weight of about 1.8 into 10 raised to power 6 Daltons. Please keep in mind the difference between the molecular weight of larger subunit and smaller subunit and how many rRNAs are present and protein or polypeptide chains are present in both the units. So, please keep this difference in your mind. So, it consists of 5 sRNA, 23 sRNA and 34 polypeptide chains. And the structure is here. This is the ridge, this is the central protuberance, this is the stalk somewhat elongated as compared to the ridge. Ridge is somewhat blunt, but it is pointed and elongated structure which is called as stalk. This is the central protuberance, this is the frontal portion, and it is the larger subunit, that is 50S. This is the back portion of the ribosome. So, prokaryotic ribosome 70S unit is here. Now, ribosomal RNA and its role. rRNA, it is transcribed from certain portion of DNA by the same energy required process for the synthesis of mRNA and tRNA. rRNA is though thought to have two roles. The 16 sRNA of the 30S subunit may aid in the initiation of protein synthesis. When the most important function, that is protein synthesis, occurs, so this 16 sRNA which are present in the 30S, that is smaller subunit of the ribosome, helps in this protein synthesis. The 3' end of the 16 sRNA complexes with an initiation signal site on the mRNA. This is all about the process of protein synthesis, which is not in our syllabus at this stage. We will study it in detail in the 6th semester, that is at your final year stage. Now, just I have to show here that these are the structures. See the uppermost diagram on right side. This is the ribosomal structure, this is the larger subunit 50S, smaller subunit 30S, and 3 sites are shown here as a pillar like structure. This is A, that is amino acyl site, this is P, that is peptidyl site, and this is E site, that is exit site, and this is the mRNA binding site. At the joining of both the subunits, this is the mRNA binding site. All these 3 sites and mRNA binding site togetherly helps in the protein synthesis chain formation or protein structure formation. So, the ribosomes have 3 sites for binding tRNA. When tRNA binds here, So these are the three sites where the tRNA binds, that is transfer RNA binds. mRNA are already itself present in the subunits, but these three sites are given here or shown here, where the tRNA comes from outside and binds here at the first stage, which is the amino acyl site or A site. Then it goes forward to the peptidyl site, and here one by one amino acids get attached here, and on the exit site the amino acid is forwarded, and from here, from the exit side, the chain of protein is given outside or processed outside. So this is all about the protein synthesis structure. See here, this is the tRNA, growing peptide chain is here, 5 prime end, 3 prime end. This is the mRNA, which is already present in the both subunits. These are the sites, and from this exit site the protein synthesis takes place, and the chain is coming outside from the another end of the ribosomes. So this is all about the now functions of ribosomes. The ribosome is involved in the process of protein synthesis. Then protein synthesis divided into 3 stages: initiation, elongation and termination. So this important function of protein synthesis is performed by the ribosomes, and in this way the main important function is performed by the ribosomes here. So dear friends, we will stop here. This is the basic ultrastructure of ribosomes, and the most important function which is performed by ribosome is the protein synthesis. So I have shown you here the ultrastructure of ribosomes, types of subunits, types of ribosomes, and how the protein synthesis takes place. How many sites are there present for the protein synthesis? A site, then amino acyl site, peptide site, and P site and exit site, that is E site. So these 3 sites and mRNAs which are already present in the ribosomes are responsible for protein synthesis, functions as protein synthesis. The stages of protein synthesis I have also explained you, that initiation, elongation and termination. These 3 stages are performed, and the polypeptide chain is formed or protein synthesis is done here. So dear friends, this is all about the ultrastructure and functions of ribosomes. Thank you very much.